Hello everyone, my name is Janet Lorv and I'm a PhD student from the University of Waterloo. And today I want to present to you my work on developing a software tool that uses reference information to defragment genes in long-read assemblies. So for our work, we decided to focus on long-read sequencing because the assemblies that were generated from these reads can produce highly contiguous sequences, unlike their short-read counterparts. But like many other researchers have encountered, these reads are highly error-prone and can have error rates upwards of 15%. And many of these errors are actually insertions and deletions, which can introduce frame shifts, as well as premature start and stop codons, leading to fragmented genes in downstream analyses. And this can even be seen in our own assembly, where 12% of single orthologs were found to be fragmented, even after multiple rounds of polishing, especially when you compare it to a short-read assembly from a closely related strain where there is no fragmented genes at all. So we decided to tackle this problem. And due to the limited amount of software tools at the time, we decided to develop our own software tool that aims to detect and correct these types of errors. Our idea was to use publicly available reference information to determine where errors are within a draft assembly. And this can be done using both reference genomes and proteins, depending on which type of data that a researcher has access to. Using three main inputs, our software goes through three main stages, starting with the error detection step, which has two methods of detection depending on the reference data that has been inputted. And this step is then followed up by an error adjustment step, where the software uses raw read data to adjust the candidate errors for strain-specific differences. After finalizing the errors, we then correct them in the draft assembly. To go into detail, our genome-based method of error detection utilizes the differences between the aligned reference genomes and the draft assemblies to detect candidate errors. We reason that the alignment differences can be indicative of a site with a potential error. For example, in this figure here, you can see every single insertion is potentially indicative of a position that may contain a deletion error. Therefore, regions with reference consensus would likely have a candidate error. We represent the frequency of these alignment differences as a calculated ADF for every single type of error and also for every position. As the software passes through the assembly, each region is then evaluated for the reference consensus indicated by a combined ADF of 1. Although there is one thing to note here is that indels greater than 2 nucleotides are ignored because we consider them to be likely strain differences rather than an assembly error. Similar to the genome-based method, our second method of detection relies on the local alignment of protein fragments to detect candidate errors. Since this method uses more distant reference information, this method is more intended for situations where there are fewer closely related genomes that could be used, such as, say, less studied organisms in metagenomic studies. The idea here is that an error is likely contained within an interfragment region for cases where nearby aligned fragments are not in agreement. The software then evaluates this region for shared nucleotides between these fragments, such as, say, scenario A, or non-shared nucleotides, such as scenario B. For example, if you look at the zoomed-in figure, the shared guanine is indicative of a potential error at that location. And how the program actually implements this entire idea is by first using the top high-scoring pair per query as a seed to approximate where the gene is, and then grouping and merging nearby low-scoring fragments on the same strand. Each interfragment region is then evaluated for a candidate error before passing on to the next step. So once we find all the error candidates, we then use raw read information to determine if a candidate has read support by using the same alignment comparisons as a genome-based detection method. If there's support, we then determine whether to retain or adjust a candidate error. This is intended to reduce reference bias by maintaining strain differences. For example, in this case here, raw read information indicates some evidence for an insertion error at the adenine rather than the reference suggested guanine. Once all errors are then finalized, they are then used to correct the draft assembly. So we tested our software on 2 draft assemblies. One of them was generated from long reads, more specifically nanopore sequencing, as well as a short read assembly, which acts as our low error dataset. For the reference information, we collected 11 equally distant reference genomes, hovering around 94 to 98% identity with our assemblies. And for the proteins, we collected approximately 5,000 ref-seq proteins from the Pseudomonas species. So after running our software on our assemblies, we were able to detect less than 3,000 errors, meaning that in all cases, less than 0.05% of the assemblies were corrected. If you look at the table with both detection methods, we were able to find substantially more errors in the long-read assemblies, with most of those errors residing in genes. However, we were also able to detect errors in the short-read assembly, but mostly they were in the introgenic region. Between the two detection methods, we were able to detect more errors using the genome-based method, mostly due to the inability to detect intergenic errors with the protein-based method. So to confirm that the corrected errors actually improved the assemblies, we annotated all the assemblies both before and after correction using NCBI's annotation pipeline. As you can see in the figure here, the overall number of annotated complete genes increased in the long-read assemblies post-correction, while the short-read assemblies do not show any noticeable improvements. In fact, there is actually a slight increase in the number of incomplete genes for the short-read assemblies, but that is largely due to a higher number of annotated genes post-correction. And if you look at the two right plots representing the different types of fragmented genes, the majority of the improvements in the long-read assemblies were from the reduction of frame-shifted genes. And with these corrections, we were able to reduce the number of fragmented genes from 23.3% to 5.6%, which is nearly comparable to the short-read assemblies' 4.4%. So we also looked into the different types of errors that we were detecting and the 4 nucleotide long k-mers that preceded these errors. And from this, we found that in the long-read assemblies, using a genome-based detection method, that the majority of the errors were insertions and deletion errors associated with long homopolymers, especially around 4 to 5 nucleotides long. And this is actually consistent with previous observations made by other researchers. And this can be actually reflective of a conservative approach in determining homopolymer length in nanopore sequencing. Substitution errors, on the other hand, did not seem to be associated with any particular k-mer and generally are not associated with homopolymers. So, in summary, we were able to develop a software tool that can use homology information from reference data to detect and correct fragmenting errors in long-read assemblies, and that correction of less than 0.05% of an assembly can drastically reduce the number of fragmented genes and improve gene annotation. With these corrections, long-read assemblies can become comparable to short-read assemblies without any additional sequencing, and hopefully, with our software, we can make long-read sequencing an attractive option to generate complete sequences in the future. Thank you all for listening.